All right, I think we'll get started. So hopefully everyone can hear me on Zoom, and then hopefully everyone can hear me in the room as well. But I hope that everyone's little reception break was nice and everyone had some great food and refreshments. And now I'm going to introduce our keynote speaker. So it's my pleasure today to introduce Dr. Megan Povolonis, an assistant professor of biology at Villanova University. Dr. Povolonis received her PhD from Johns Hopkins University School of Medicine, and then she went on to do a postdoc at Oxford. She worked at Penn State Brandywine campus as a professor and then began her career at Villanova in 2020, actually during the height of the pandemic. Her lab studies parasite vector interactions and mitochondrial morphology in kinetoplastid parasites. Dr. Povolonis is also an incredibly dedicated teacher and mentor and has been recognized for her impressive efforts to mentor numerous trainees. So we're really excited for Dr. Povolonis today. Thank you for being our keynote speaker, and we can't wait to hear about both your research and your academic journey. So without further ado, let Dr. Povolonis share her story. Thank you.
Oh, yeah. Okay, thank you so much, Marissa, for that very kind introduction, and thank you for the invitation to be here. I'm very excited to be here to talk about my work. It's always and a special compliment to be invited by students, so I'm really delighted by that. So I'm going to talk about today this unusual little parasite, Chlamydia fasciculata, and hopefully it'll become clear why I'm interested in this organism. And yes, as Marissa said, I was asked to give a few slides about kind of my academic journey and how I ended up where I am. So because I teach undergraduates, I thought it would be good to start with my undergraduate research experience. So I went to Loyola College in Maryland, which is now called Loyola University. And so that's where I was really introduced to this model of liberal arts education. I was interested in science, but I was also interested in a lot of other things. So that was a pretty good fit for me. And because I was interested in so many things, I didn't really think about the idea of doing research until relatively late. So as a junior, I kind of walked into the office of my favorite biology teacher and said, yeah, so I want to like work in your lab, thinking that I was making a really good offer to work for free in the lab. And she said, yeah, that's great. I don't really have any time to supervise you right now. And now, as someone who has to tell really talented students no all the time myself, I kind of understand exactly why sometimes it has to happen. But at the time, I was really kind of shocked, like, oh, okay. But she helped me out because she knew of someone else who might be looking for an undergraduate student. And that was Dr. Erica Matunis, who worked down the road at the Carnegie Institute, Department of Embryology, which is this little unassuming little building here tucked into the corner of the Homewood campus, which has since been torn down. But it was really chock full of amazing scientists. And I'm kind of glad that I was too naive to understand some of the people I was working alongside. I probably would have been so intimidated that I was non-functional. But Andy Fire was doing work there at the time, who went on to win the Nobel Prize for discovering RNAi, and C. elegans. And so it was a really kind of special research opportunity to work in Erica's lab. When I interviewed with Erica, she only had 2 questions for me. She said, can you show up when you say you're going to show up? Which again, as I do research with undergraduates, I now kind of understand the background to that question. And the second question was, if I give you 10x PBS and I ask you to make 1x 1 liter PBS, can you do it? How would you do it? And I was able to answer that question, thankfully. And she was like, okay, I'll teach you the rest. So I think of that also when I'm interviewing undergraduate students. And I think that's part of the reason why I do what I do, is that, you know, someone has to give every scientist their first research experience. So someone has to be willing to take on the student who knows literally nothing. And that's what I get to do. And it has its challenges and it has its rewards as well. So Erica studied stem cell differentiation and how the niche of stem cells in the testis, how signal transduction kind of established stem cell fate. So whether a cell became a stem cell or differentiated into a sperm cell. And so I got to learn a lot about the Drosophila testis and fly pushing and fly genetics. And it was just a really great experience. So then I went on, I stayed in Baltimore to do my PhD at Johns Hopkins down at the School of Medicine. And when I started, I thought I wanted to be a virologist. So I did 2 rotations in very, very excellent virology labs. And then in my third rotation, someone said, oh, you should try out this parasite lab because the mentor is really, really good and the lab has a really good vibe. So you should try it out. Something different. So I met with Dr. Paula England, who was a very preeminent parasitologist at the time. He studied these little parasites called trypanosomes, so Trypanosoma brucei, and I'd never heard of them before. I knew nothing about single-celled parasites. But even from our first conversation, just learning about their amazing molecular biology, their amazing cell biology, their incredible complexity and weirdness, I was pretty much hooked. So then sorry, virologist, I forgot about viruses, and I have been working on these organisms ever since. I've been stuck on eukaryotic parasites for my entire career. So what I studied in Paul's lab was the replication of their mitochondrial genome, which is very unusual. So you can see that when you stain trypanosomes with the DNA stained DAPI, you can see two structures. You can see the nucleus, and then you can see this brightly staining dot, which is called the kinetoplast DNA or kDNA. So this is what gives this group of parasites their name, the kinetoplastids. And it's called a kinetoplastid because it's kind of near the basal body of the flagellum. So people thought it was involved in movement. It really doesn't do anything with movement. It's the mitochondrial genome. And it looks like this when you see it in a TEM. But then when you isolate this structure and you deprotonate it, it spreads out, and you can see that it's this incredibly elaborate network of circles. So topologically interlocked, thousands of circles. There's larger circles that are the main mitochondrial genome. There's these smaller circles called mini circles. And this whole structure has to replicate with relatively high fidelity once and only once per cell cycle, because there's only one kDNA per cell. So Paul's lab spent about 40 years of continuous NIH funding studying the mechanism of kDNA replication, and I was able to do a little part of that as part of that work in his lab. So as a little aside, Paul was very supportive of sending me to a residential course at the Marine Biological Laboratory in Woods Hole. If anybody, trainees, if you ever get a chance, if anybody's ever willing to send you to Woods Hole to one of these courses, you should definitely do it. They have a bunch of different courses on, a lot of neuro stuff, but also parasites, fungal pathogenesis, embryology, all of these courses. As you live there, and 6 days a week, we had lecturers flown in from all over the world to teach about their research. And then in the afternoon, evening, and late into the night, we did experiments. So this is a transformative experience for me for a couple different reasons. So first of all, it gave me a lot of confidence that that I could learn new techniques, that I could learn new systems. We were working with a different parasitic species every 2 weeks. And so at a really critical time in my graduate career, this is exactly what I needed. This is the course that really convinced me that I wanted to stay in parasitology, because again, after I saw all of the other amazing things you can do with parasites, I was really excited to kind of keep going. And on a personal note, if you can see these two fresh-faced young people sitting next to each other on the steps here, this is where I met my husband and current collaborator. We met at this course. So at the time, he had decided he was going to do his postdoc in the UK. And since there are a lot of great parasitologists working in the UK, I decided, sure, that sounded great. I was going to go to the UK too. And so that's how I ended up in Gloria Rudenko's lab. So Gloria, when I first started working with her, I was at the University of Oxford. While I was there, so she moved her lab to Imperial College London, where I got to work in this cool architectural landmark, the Sir Alexander Fleming building, which is a really cool place to do science. But Gloria was interested in this other process that trypanosomes are famous for, antigenic variation. So trypanosomes can change their surface coat in order to evade the adaptive immune response. And there's a lot of epigenetic mechanisms involved in this, because there are thousands of surface coat genes and only one is expressed at a time. So this is a system of monolithic exclusion, and Gloria's lab studied that. So the model of this is briefly is simplified, is that you have a bunch of trypanosomes, if you get infected, that all express the same VSG, the same antigenic determinant VSG. Your adaptive immune system does what it's supposed to do. It makes antibodies that recognizes that surface coat protein and kills off those trypanosomes. But in the meantime, a certain small percentage of them have switched to expression of a new VSG. And this process, as it continues, results in waves of parasitemia. So this is a phenomenon that's been noted in cases of African sleeping sickness for over 100 years, and it's caused by antigenic variation. So this is a fascinating system. Again, it's a very, kind of active, competitive, field in trypanosome biology. I learned a lot of great molecular biology in Gloria's lab, but when I was going to start my own lab, I knew I wanted to be a teaching-focused institution. I really had valued that as part of my own training. I knew I wanted to be in an undergraduate-focused institution, and I really didn't think that this was going to be the field where I was going to be able to run the kind of lab that I wanted to run and still be able to make contributions and continue to publish and continue to do research. So I made a big switch to a completely different organism. So this is Chrysidia fasciculata, which is a parasite that's related to trypanosomes, but it doesn't infect humans, it just infects mosquitoes. So it's a mosquito parasite, and the reason I knew about it was that in Paul's lab, in my graduate work, they occasionally would use this organism as a biochemical model because you can grow ridiculous quantities of them, 10 to the 8th per mil, in media that doesn't require any serum. So for a eukaryote, this is incredibly powerful. So if you want to do kind of, old school protein purification, you want to purify an activity, a replication, enzyme, primase, polymerase, something like that, you can grow up 40 liters of these guys, and it's not going to cost you that much money. They can grow at room temperature. They're very robust. So occasionally I would see people growing these things, and I thought, yes, I'm going to work with undergraduates. This is a BSL level 1 organism. It'll be really cheap and easy for me to grow. And I was kind of naive about it because I thought, oh yeah, I'm sure they're the same as trypanosomes. I'm sure I'll be able to learn this new organism, no problem. And of course, it hasn't been quite that easy, but I still, I've been able to learn a lot from Chrysidia fasciculata. So here's just what they look like in culture. And we can also, even the cultured strains can still infect mosquitoes, so we can infect laboratory mosquitoes. So this is a really nice model because we can actually study them in culture, but they still have the ability to infect their natural host. So this is just to say, I have a lot of fun in my job, blending teaching and research. It's a little bit different from kind of the environments that I was trained in, which were very R1, research environments. And I would say, the blend of teaching and research has changed over time. Some years I've been 60% teaching, and some years I'm 60% research. But it's nice that I can continue to explore, what it means to be at the interface between these two. Okay. So now to get into a little bit of what I've been doing with Chrysidia over the past few years, I first want to give an introduction to some kinetoplastid parasites that you may have heard of. I'm just assuming that nobody's heard of Chrysidia because that's true even in a lot of parasitology audiences. So most of the time when you hear about these parasites, you're talking about the big 3. So these are the big 3 disease-causing kinetoplastid parasites. They all have very similar structure. They're single-celled eukaryotes. They have a flagellum. They swim around. They're usually transmitted to us by insects. So the organism that I spent all of my training on was Trypanosoma brucei, which is transmitted solely in Africa by the tsetse fly. We have Trypanosoma cruzi, which is transmitted in South and Central America by the triatamine bug, also known as the rejuvid bug or the assassin bug, this nasty looking guy here. And then finally, probably the one that affects the most number of people in terms of cases per year is leishmaniasis. Leishmania parasites, there are several species that cause disease. There's a variety of clinical manifestations depending on which species you get infected with, and they're transmitted by sandflies. So kinetoplastids, we care about them because they infect people, but they're also important veterinary pathogens. So there are other subspecies of Trypanosoma brucei and other species related to T. brucei that cause disease in domesticated cattle in Africa, which severely limits economic development in these regions. There are broad regions of Africa where you can't raise cattle because of these diseases. And also, if you're interested in bees at all, which a lot of us are these days, there are kinetoplastid parasites that solely infect bees, and they do actually cause some ill effects. So this is not kind of the sole cause of pollinator decline, but this is one contributing factor that people are looking into as possibly contributing to a reduction in pollinator health, particularly for bumblebees and and honeybees. So you can see here we have a parasite that's adhered to the gut of the bee host, and I'll talk more about that later on. So this adhesion phenotype is one of the first things that I noted when I started growing Ophidia fasciculata in the lab. Again, I'd never grown them before. And reading the literature, I found that they said that you needed to grow your cultures on a rocker. Otherwise, they would stick to the plastic and they would grow as this adherent form of parasites. And I found this wild because trypanosomes don't do this. I'd never encountered this before in growing Trypanosoma brucei. Not only that, we could actually watch this. So this is a time-lapse series under the microscope done with our collaborators at Penn, where we can actually watch single cells sit down on the surface. They transition to an adherent cell, and then they divide as attached cells that are attached to the surface. And this really raised a lot of questions for me. So why do they do this and trypanosomes don't do this in culture? How do they know where to sit down? And then how do they know that they've sat down and that now they're going to divide as an attached cell instead of, forming a swimming cell and swimming away? Because you can see occasionally some of these cell divisions do produce swimming cells, but then go swim away and maybe even can go sit down somewhere else. So I thought this whole behavior was really curious, and I didn't know if it was an in vitro artifact or something. But it turns out, as I, again, learned more about the biology of this organism, so this is a mosquito parasite. And this attachment is actually critical for the parasite to effectively colonize its insect host. So we're looking at a mosquito cut and cross section here. The yellow is the gut. And if we pull it out and dissect it, we can see this here. So what happens is if the mosquito takes a sugar meal that's containing parasites, they first go to this organ indicated by the pink arrow called the crop. So this is where you get some of the water absorption coming through the crop. And then eventually the rest of the meal goes to the midgut, and then finally to the hindgut and the rectum. And it's those last two tissues where you really see accumulation of the parasite. And the parasites are actually attached to the lining of those tissues, where they grow as these adherent rosettes, which really look a lot like the rosettes that we observe in culture. So this is where I learned that, oh, this is not some weird in vitro artifact. This is actually a part of their life cycle that maybe I'm able to replicate in culture. And again, I wasn't the first person to take note of this, but I did find it interesting. One of the reasons I found it interesting is because, when you study parasites, one of the main parts of studying parasites is that they undergo these really dramatic developmental transitions as they go through different environments. A lot of times, especially if they have to adapt to different hosts, they have to change form, change metabolism, change their morphology in order to survive in those host niches. And even though I've been studying trypanosomes for a long time, I had never known that even Trypanosoma brucei shows this behavior as well in its tsetse fly host. So most of the stages of T. brucei in the mammal and in the fly are free swimming, again, with their single flagellum, but there is an absolutely required stage for the parasite to adhere via its flagellum to the salivary gland epithelium. And from there, it can transition to this metacyclic form, which is the infectious form. So this is the form that actually gets in you and infects you when you're bitten by a tsetse fly in Africa. And again, upon reading a little further, this is not restricted to Trypanosoma brucei. And here we're looking at kind of the ultrastructure of the attachment itself. Again, it forms in the flagellum, and it's this electron-dense kind of plaque that looks a lot like a hemidesmosome, although the proteins that make hemidesmosomes are not really conserved as far as we can tell in these organisms. So this is what it looks like when the flagellum of T. brucei attaches to the salivary gland. In T. cruzi, you can see the same thing happening to parasites that are attached via their flagellum to the hindgut of the rejuvid bug. And even in Leishmania, it's kind of unusual. Where they attach is the stomoidal valve of the sandfly, which acts a little bit like an epiglottis. And it's thought that actually this attachment facilitates transmission of the parasites because it compromises the function of the valve, so that infected sandflies are more likely to regurgitate while feeding, which means that they expel more parasites into the bite wound than they normally would. So this is interesting because in all of these species, the ultrastructure, the morphology of the attachment is similar. It always uses the flagellum, but the tissue that it's attaching to is completely different. So there are lots of questions that are outstanding about adherence of kinetoplasma to insect tissues because, especially for these pathogenic species, these developmental stages are kind of hard to access. So they only really occur in the insect for the most part. Insect infections for these particular insect colonies can be difficult. It's difficult to get numbers of that developmental form, to do kind of large scale analysis. It's difficult to get the timing of the infection right, to kind of find these developmental forms at the right time. So even though this had been kind of described with EM and kind of in the literature, we didn't really know a lot about the mechanism of how it happens. So the major questions that we had on initiating this project were, what are the molecules that actually allow the kinetoplastid, the parasites, to attach? So what proteins are there making up that hemidesmosome-like structure? Why do kinetoplastids adhere to particular tissue? So why are some on one tissue and some are another tissue? What signals are they responding to, and what tells them to attach at a particular place? And so that really made us think about that signal transduction must be involved in this process. There must be something that they're sensing, and that triggers them to transition to a new developmental form, the attached form. So where we thought the Bacteria fasciculata might be powerful is this in vitro attachment phenomena. So the fact that we can get parasites to attach to plastic, and we can get them to do that in a reasonable in number. So pretty much we can generate as many attached parasites as we give them plastic. We can get monolayers of Bacteria. So this is nice, but of course we wanted to make sure that what we were looking at in vitro kind of approximated what we were looking at in vivo. And so we did some electron microscopy of cells that were attached to plastic to compare that to what we knew from the literature of parasites adhered in the mosquito. And what we saw was pretty similar. So we see here, here's a parasite, and it has a shortened flagellum. And there is an electron-dense plaque here where the tip of the shortened flagellum contacts the plastic substrate. So this was also done in collaboration with the EM facility at Penn that helped us out with these. And so we thought, well, it may not be exactly the same as what's happening in vivo, but at least the structural components are there, and some of the other factors, the filaments that are in this attachment plaque are present in adhered cells in vitro. We could also, through the time course studies like that movie that I just showed you, we can kind of define the stages of attachment in a way that's very difficult to do with those pathogenic kinetoplastids where they're only really doing it inside the insect. So what we could see from this is that you would have a cell that is swimming around happily in the culture that would encounter the dish, and it usually encounters it via this part of the cell body where the flagellum exits the cell. This seems to trigger shortening of the flagellum, and then eventually formation of the hemidesmosome-like plaque between the tip of the flagellum and the substrate. Flagellus shortening we could measure. So we could look at the kinetics of flagellus shortening. It's actually quite rapid from the point at which they contact the dish. In about an hour and a half, they've completed flagellus shortening, and then essentially they just look like little circles that are all attached to the substrate via the shortened flagella. We also developed an attachment assay that allows us to quantitate the amount of attachment that's happening under various conditions. So to do this, we can allow the parasites to contact the plate. We can wash away anybody that doesn't stick. And then we can count how many parasites have adhered after 2 hours. And then we can let them grow for 24 hours and form these attached rosettes. And then we can count how many rosettes we get at the end. So this is a really nice way to get quantitative data, not only at the rate of attachment, but also their ability to grow as attached cells. We could also do fun microscopy. So the blue is DNA, so that's mainly the nucleus that you're seeing, and the green is a flagellar marker. And so this way we can look at the morphology of the cells of the rosette to describe the fact that each cell is contacting the substrate. So at least so far, we have not observed any contact of the cells with each other. Each cell in the rosette is reaching down with its own flagellum to contact the substrate. So we don't see any cell-cell connections, at least that we can see so far. So it really is, you have a cell that sits down and divides to make a rosette of cells that are all individually attached. because we could enrich for swimming and attached cells in culture, we could also do RNA to look at gene expression differences between these two different forms. So when we did this, we found actually a pretty large number of changes between swimming parasites and adhered parasites. And this maybe should not have been so surprising, but it was a little bit surprising to me. It does reinforce the idea, I think, that these are really two distinct developmental stages, the swimming and the attached form. What was surprising to me about it was they're in exactly the same media, at the same temperature, under the same conditions. Nothing has been done to these cells except that the adherent ones have been allowed to attach to a surface and allowed to grow as attached cells. So that's really the only difference between the two. And in other kinetoplastid models, usually when you want to study differentiation, you have to do some kind of perturbation. You have to give them a chemical or give them a temperature shock or a pH shift or something like that, osmotic shock, to get them to differentiate. And here it really seemed like surface contact was driving major changes in gene expression. So then, of course, we wanted to see what some of these changes were. And one of the strongest signals that came out of this was in the swimming cells, where we saw a lot of genes whose products have predictive roles in signal transduction. So there were a lot of signaling proteins that came out of this, but one of the biggest groups were proteins involved in cyclic AMP-based signal transduction. So cyclic AMP signaling is obviously a universal second messenger found in pretty much all organisms. So we could fill a whole class on all of the things that cyclic AMP does in different organisms. It does play a role in adhesion and biofilm formation in bacteria. It's very famous. There's beautiful movies you can look at on cyclic AMP propagation waves in dictyostelium. It controls development and chemoattraction in dictyostelium. Even in trypanosomatids like T. brucei, it has been implicated in differentiation events, so developmental transitions from one stage to another. It's also involved in this really cool process called social motility. So this is in Trypanosoma brucei. There's a cool assay for this, where if you dot, these are actually parasites, and it's pretty hard to see on the screen, but if you dot Trypanosoma brucei parasites in the middle of an agar plate, they will send out these spokes, right, reproducibly. Then there's all kinds of cool, weird things where the spokes avoid each other, and then if a parasite gets lost off the spoke, his friends will go and pick him up. And so there are things going on that they call social motility, where the parasites are aware of each other, they're sensing each other, they're having this coordinated movement. And people have done a lot of work on this to show that this is actually how the parasites find their way through the tsetse fly. They have to undergo this complicated migration from the gut to the salivary gland. And if you mess with cyclic AMP signaling, they lose their ability to form spokes in this assay, and they also lose the ability to migrate in the fly, even though they have no motility defect and no growth defect. It really is that they can't sense and respond to their signals appropriately, and they can't do this collective movement through the fly. So there was a reasonable amount of data already on cyclic AMP signaling, even in trypanosomatids, and we wanted to look at it in the context of attachment. So just a brief primer on cyclic AMP signaling. This is, of course, through a eukaryotic lens, but most of us probably learn about the signaling pathway as a ligand binds to a G protein coupled receptor, which activates a heterotrimeric G protein, which activates adenylate cyclase, which converts ATP to cyclic AMP, which can then signal to downstream effectors, usually protein kinase A, which can then phosphorylate targets and lead to a cellular response. The signal is terminated by phosphodiesterases, which can degrade the cyclic AMP and turn the signal off. Kinetoplastids, of course, always have to do things a little bit different. There's no GPCRs, there's no heterotrimeric G proteins. They have a bunch of these adenylate cyclases that have things that look like ligand binding domains on them. So they look like they could function as receptors, although no one has yet found a ligand for any of these. So there are these receptor adenylate cyclases responding to unknown signals. They do make cyclic AMP. The protein kinase A in kinetoplastids does not seem to be dependent on cyclic AMP for regulation. So the downstream effectors in this pathway are also not known, but they do have phosphodiesterases that acted the same way. So our next question was about, okay, so let's look at some of these signaling components and where they're localized in the cell, because in the Trypanosome brucei studies, when they were looking at social motility, they actually found that a lot of these signaling proteins were found in the flagellum. So we localized one of the Crispidia receptor adenylate cyclases and one of the phosphodiesterases, and they were both found in the flagellum. So this is PF16. This is a central pair marker, just showing us where the flagellum is. And here we can see that the PDE is localized along the length of the flagellum. However, the receptor adenylate cyclase is found primarily at the flagellar tip. So this is interesting because the flagellum obviously has many roles in these parasites. So it's obviously functioning in motility, but it also may be functioning as a kind of sensory cilium, a place to organize signal transduction to allow the parasite to know where it is and respond to its environment. In fact, I know one prominent flagellar researcher that calls the flagellum the nose of the parasite. And that's because kinetoplastids, I don't know if you're familiar, they actually swim backwards to what you would think. So, you know, when you think of these things with the tail, you think that it's a tail, and they swim this way, right? The flagellum actually rotates like a corkscrew, and they actually swim towards their flagellum. So when you're looking at the tip of the flagellum, that's the anterior end of the parasite. It is the nose, and this is the posterior end. So a little bit backwards from what you would expect. So we were excited to see that even in Crispidia, some of these signal transduction proteins were found in the flagellum, as they had been shown in other kinetoplastids. But interestingly, when we tried to localize these proteins in attached cells, we didn't see any signal in the flagellum at all. Now, the flagellum is still there, as you can see from our PF16 control. It's shorter, but it's still detectable. But we never found any signal in the flagellum for either the PDE or the RAF1. So I think this is really interesting because it implies that there's some kind of different protein localization that happens during this developmental transition from the swimming to the attached. So we're still doing some more confirmation on this to kind of see where these proteins go. We can still detect them by Western blot. So you can see that the RAC and swimming in adherent cells is still detectable. The PDEA does seem a little bit less abundant in the attached cells. But we don't see any signal in the flagellum. So we're interested in kind of how proteins get localized to the flagellum and how this is different in these two different forms of the parasite. So next we want to see if we could manipulate this pathway and if that would mess with adhesion at all. So kinetoplastids have a lot of these RACs. So trypanosomes, T. brucei actually has a ton. They have about 60 receptor adenylate cyclases. Crispidia has fewer. I have about 9, and they have about 5 PDEs. So we started with a chemical inhibition strategy. So there was a compound that had already been developed as a specific phosphodiesterase inhibitor for kinetoplastid PDEs, and it was developed actually as a drug, a potential drug to treat sleeping sickness. So what happens is this compound MPD1 inhibits phosphodiesterases, which should lead to an increase in cyclic AMP. So we exposed swimming cells to this compound, and then we performed our standard adhesion assays. This is the one where we allow cells to attach, and then we wash away unattached cells, and then we count how many cells have attached. And this compound MPD1 dramatically blocks attachment. So every single assay that we've done, the presence of the compound interferes with attachment. We didn't see any attached cells, and this is a dose-dependent response. And also we found a related compound, MPD226, that had the same response. So when we are inhibiting a phosphodiesterase and increasing the amount of cyclic AMP, we're doing something that blocks adhesion. So of course, we wanted to make sure that the compound wasn't simply killing the parasite. So we did a growth curve. We're exposing swimming cells to MPD1 and then just monitored growth. And as you can see, swimming cells grow quite happily in the compound. So they grow, they're morphologically, as far as we can tell, indistinguishable from non-treated cells, but they've lost the ability to attach. Surprisingly, adhered cells also can grow perfectly fine in the compound. So if we allow the connection to establish in that initial stage and then put the compound on, they will continue to divide as attached cells just fine. So this was really surprising to us, but it has contributed to our model where we're thinking that messing with cyclic AMP is actually affecting the transition from swimming to attach. Once they're stably attached, once they've adopted that cell fate, the changes in cyclic AMP flux don't seem to affect them. So next, we wanted to make sure that the compound MPD1, even though it's very well characterized in Trypanosome abruzii, was actually doing the thing that we thought it was doing in Crithidia. So in collaboration with the Wistar Metabolomics Core, they were able to measure cyclic AMP levels in our MPD1 treated cells. So here you can see that compared to vehicle treated cells, cells treated with MPD1 have increased cyclic AMP levels, whereas levels of another metabolite, AMP, were unaffected. And here this is just showing that the MPD1 compound is in fact getting into the cells, so we could also measure that. So next we wanted to take a genetic approach. So there are some genetic tools that are available in Crithidia fasciculata, not many, not as many as in the much more widely studied trypanosomes. But we can do kind of traditional knockouts. So we can make constructs where we have regions of homology flanking a drug cassette that match the gene that we're trying to knock out. These are diploid organisms, so we have to, in successive rounds of transfection, knock out both genes. So we can replace the first gene with a hygromycin cassette and the second copy, the second allele with a neomycin cassette. And we were able to do this for one of the phosphodiesterases, one that had been very well studied in Trypanosome abruzii. And we found that, again, when we measured the attachment frequency of our mutants, that they were dramatically impaired in attachment. So you can see the heterozygote is slightly less frequent in attachment, but two individual clones of knockout are less able to attach. So this also fits with our MPD1 study. So MPD1 is an inhibitor of phosphodiesterases that blocks attachment, and it seems like genetic knockout of one of the major phosphodiesterases also inhibits attachment. We were also able to make a heterozygote knockout of one of the racks. So the heterozygote actually has a pretty significant growth phenotype, so we haven't tried to knock out the other copy. We have a sense that maybe that's not going to go so well. But interestingly, when we use the attachment assay to measure the attachment frequency of the RAC1 het, they have the opposite phenotype. They actually have an increased attachment phenotype. So if you have knockout of a rack, so half the amount of the rack. So this is the same rack that's at the very tip of the flagellum. If you knock out one copy of that, presumably decreasing cyclic AMP, they actually increase in attachment. Another interesting phenotype, it's really hard to see, sorry. Another interesting phenotype of these RAC1 heterozygous cells that we don't quite understand, but we're interested in following up on. So this is just to remind you that this is the rack that's found at the tip of the flagellum. Their flagella is slightly shorter, very slightly. So my Kelly Hodges, a talented master's degree student who recently left my lab, actually measured flagella in the parental and the heterozygous knockout cells and found that the flagella was significantly shorter. It almost looks like, and again, we have to confirm this with the markers, like they're missing that tip of the flagellum where the RAC1 was localized. So we're not really sure what that means, but we think it's an interesting finding that maybe there is something structurally required for the flagellum with these cyclic AMP signaling components. So another kind of technology that our collaborators at Penn have been working on in order to attack the in vivo side of these, the behavior of these parasites, is a way to better quantitate how many parasites are in the natural host, the mosquito. So of course, it's really nice to do all these tests of like attachment frequency in vitro. It's very quick and easy to do. We can make it very quantitative, and we can look at all the various stages of attachment and generate loads of material. But we'd like to see how relevant this is to actually the life cycle of the parasite in their host. So in order to do that, we have parasites that express nanoluciferase. And so we have a pretty good linear range of detection for both swimming and attached cells in culture. And then we can also just infect mosquitoes with these parasites pull out the various tissues and pop them in a luciferase assay, and then quantitatively determine how many parasites are in there. So counting this with GFP by hand or even with ImageJ might have been a little bit challenging. So we're hoping that this luciferase assay will allow us to actually test some of these mutants. So now we have these mutants that we've generated, we can introduce luciferase and see if they're compromised in their ability to infect the mosquito, or in fact, if they're better at infecting the mosquito. So here's our model for how the cyclic AMP signaling might be working. Again, we have a lot more work to do in this area. But again, we really think that there must be a PDE-dependent drop in cyclic AMP that happens specifically when the cells are transitioning. Now, we don't really know what might be causing this. We're intrigued by the idea that this initial attachment is somehow mechanically sensed. It does seem to trigger shortening of the flagellum, which could change the localization of some of these signaling proteins and affect cyclic AMP flux in the flagellum. And that this somehow leads to first an initially attached state, which is then later reinforced to become the fully attached state, complete with the hemidesmosome. So if you treat cells with MPD1, they have a low probability of making this transition. The PDEB1 mutant also has a low transition probability, because it has higher cyclic AMP levels to begin with. The RAC1 mutant, because it already has lower cyclic AMP levels, has a higher transition probability because it's already kind of closer to that threshold for that cyclic AMP dip that we think drives the differentiation. So in the last little bit of the talk, I just want to talk a bit about the mosquito. So again, we had these pictures where we could see GFP expression of parasites, and we could actually trace in which tissues the parasites were. And again, they're primarily in the hindgut and the rectum. And we were really struck by how infected these mosquitoes are. They're really teeming parasites, and 100% of our mosquitoes get infected every time. This is like such a reliable system. They really love infecting mosquitoes. And this is unheard of kind of in the world of Tsetse and other things where they try to generate these infections. They're really not this robust, but Graphithia really wants to grow in a mosquito, and it's very easy to get lots of infected mosquitoes. But despite being full of parasites, the mosquitoes are really unbothered by this. So we didn't see any negative effects from being an infected mosquito, no effect on lifespan, no effect on egg laying or any of those. Now, we haven't particularly stressed them to see if that causes an effect, but essentially these parasites may just be a commensal for the mosquito, just part of their normal microbiome. Similarly, we did do dual RNA-seq, so we actually infected mosquitoes with parasites, and then we compared infected and uninfected mosquitoes to see if there was any difference in gene expression, and there's not a lot of changes, as you might expect from the fact that the mosquitoes seem relatively unbothered. There are a few changes that are intriguing, perhaps in some things in the innate immune pathway of the mosquitoes, so we are interested in kind of, if the mosquito is responding to the presence of the parasites in the gut, even if that's not a particularly dramatic response. And one of the reasons why we're interested in this is that there are Graphithia parasites that do have major impacts on their insect hosts. And that's where I'm going to finish up this talk with talking a little bit about our work on bee parasites. So we have a new project in the lab where we're actually working with Graphithia bombi, which is a closely related parasite that infects bumblebees. And it has a very similar life cycle in the bee as Graphithia fasciculata has in the mosquito. So this is a diagram of the fecal-oral transmission route of these parasites. So an infected bumblebee will deposit in its feces parasites on a flower, and then the next bee comes along and takes their nectar and pollen meal and will infect itself with parasites. The parasites go to the hindgut where they adhere via their flagellum in the same way as Graphithia fasciculata, except in this case, infection seems to cause decreased fitness and particularly decreased reproductive capacity of queens. So queens are much less likely to be able to found a new colony the next year if they're infected. So our collaborators have found this really interesting phenotype where if bees are fed sunflower pollen, they can cure their parasitic infections. So we're all interested, I think, in how we could make better pollinator habitats and healthier bees, and what do I plant in my yard to make the bees happier. And there's a lot of, kind of big organizations that are interested in investing in pollinator habitats. So my collaborators are really kind of plant biologists who are interested in kind of how do we structure pollinator habitats to better support pollinator health. And they're interested in Crithidia bombi as a model of a host pathogen interaction. So they discovered the specific effect of sunflower pollen and other Asteraceae pollen in preventing infection of bees by Crithidia bombi and clearing existing infections. And this is just some really cool pictures. Pollen is really pretty, and sunflower pollen in particular has these like cool little aggressive spikes coming off of it. So other pollens don't have that kind of morphology. And this was actually taken in my lab. This is what the pollen looks like under the microscope. So people send me pollen now and I have it like in my freezer so I can throw it on my parasites and see what they do. So right now, in this project, we're trying to apply the same things that we've learned from Crithidia fasciculata to Crithidia bombi. So we've done what we think is the first ever transfection of Crithidia bombi. So we've been able to make fluorescent Crithidia bombi. We've now put nanoluciferase in Crithidia bombi, so we can actually track infections of laboratory colonies of bees. We can actually do that in the presence of different pollens to maybe see like when the parasites kind of are killed, or when, in what tissues they end up in, when that infection, kind of breaks down. We can do, so this is in Crithidia bombi, we can do in vitro adhesion assays, just like we did with Crithidia fasciculata. So we can test different pollen components and see, do they affect growth, do they affect morphology, do they affect adherence. This is a really exciting kind of application of this work. And I never really anticipated that I'd be working with ecologists, but it's kind of fun. And it's also really nice to kind of recruit some students to my lab who have a real interdisciplinary focus. They're interested in ecological real world questions, and as well as some cell molecular biology also. So in summary, I think Crithidia fasciculata is a pretty fun model organism, and it's particularly well suited to studying kinetoplastid attachment to insect tissues. We think the cyclic AMP signaling is definitely involved in the transition from swimming to attached. We can look at the role of signaling compounds in attachment, both in vitro and in vitro and in vivo in this system, which I think is really helpful and is kind of unique in kinetoplastids. And we hope that our findings are going to provide insight to how pathogenic kinetoplastids attach their insect hosts, and also some of these pollinator pathogens as well. So it's a very crowded acknowledgement slide because I collaborate with a lot of folks. So most important is the POVE lab at University of Pennsylvania. So ongoing collaboration there, where we do all of our mosquito work and a lot of the microscopy, also imaging with Star Institute, all the wonderful people in my lab. So Maddie, Andre, Kelly, Nikki, so all of this work was done by either undergraduates or master's degree students or technicians. That all have driven this project from the beginning, and our funders. And thank you so much for your attention.
Thank you so much. So if anyone has any questions for Dr. Pavon, this is really great talk.
I think I can speak loud. I was just wondering whether fasciculata would also serve, infect your bumblebees and serve as probiotics for against the bombi, whatever.
Yeah, that's a great question. So we haven't done those experiments, so we haven't tried to see if fasciculata will infect bombi. So far, I think people have only found it in mosquitoes. So it may not be well suited to the bee gut. We've actually thought about, what if we could do something to the fasciculata that would mess with mosquitoes? That's something that we've thought about doing, but
it sticks to plastic.
It sticks to plastic. Yeah, so why doesn't it infect the bee? It's a very good question. It's why don't you find it in anything, any insect? So I would say every insect seems to have its kinetoplastid. So pretty much even if you went out here and you found any old insect, black fly, whatever, you pulled it open, you probably have a 90% chance of pulling out some kind of kinetoplastic parasite. Whether it's something known to science or not is not clear. There's a person in our field that just travels around the world pulling apart bugs and describing these species. Sounds like a pretty good deal to me. Picks really nice locations to look for them too. But yeah, the species specificity, I mean, in leishmania, it's really specific. Which species of leishmania can be in which sandfly? And I think you know, for Perfidia bombi, I know they can also infect honeybees, but I don't know about the barriers, which is a really interesting question. Yeah.
Is anything known about the first step of binding, which is flagella independent?
Yes, that is a great question. We would love to know more about the first step of binding and what makes a cell competent to adhere. The short answer is no. In C. cruzi, there's some evidence that osmotic stress might, so if you osmotically stress the cells, so you can do some T. cruzi type adhesion assays as well. And osmotic stress seems to make them more likely to adhere. But that was all done in vitro. We haven't done that yet in ours.
So you get the adhesion when you put your parasites in them, just like a plastic plate. What if you just mix beads in with your culture?
Ah,
You know,
yes.
is that enough to make them, you know, they wouldn't, will they clump around your beads?
They will, yeah. So we haven't done that, but there's literature where they will stick to beads. We've done some things like, you know, for instance, they stick better to tissue culture treated plastic than to untreated plastic. They stick better to plastic than they do to glass. So, and we haven't done it. I mean, people have tried to stick them, I think, to millipore filters and beads. So yeah, I think they absolutely would stick to beads. So I think people have even tried to use that, maybe even in the bacterial field, to kind of purify attached
Well,
cells.
I was thinking when you said the thing about like maybe sensing kind of mechanical stress, there are really cool T cell receptor papers where they show that like MHC coded beads bind to TCRs and then they can trap the beads with a laser and tug on them. And it like enhances the affinity of the TCR MHC interaction. So that would be, I don't know how feasible it is. But
Yeah, no, that would be really cool. Yeah, I think the mechanical stress thing, I think, is a really interesting, because again, we don't know if they just like randomly encounter the surface, but then they're kind of like stuck and then they're sensing that kind of tension, or there's something that messes with their, you know, that particular region of the cell, and then that kind of triggers the whole process. What's interesting is that in the MPD1 treated cells, we've done some live cell imaging and we do see adhesion failures. So we see them like they get stuck initially, but they can't transition. So then they just swim away. We haven't finished analyzing all that data quantitatively. This is why I didn't show it, but we really think that, yeah, maybe that initial stage is kind of a physical stuck, but then that triggers the transition. But if they can't do the signaling, then they can't stay there.
And when you do your like comparative RNA-seq with the swimming and the attached, are you swimming the ones that you rinsed off, or are they just ones that you didn't attach at all?
No, they are just the ones we didn't attach at all. They're not the ones we rinsed off. And I think that is a really interesting question, because we've often thought of, yeah, again, I think there's literature in the biofilm field for this too, like if they have some kind of memory, like if they're freshly divided off, you know, are they more likely to stick the next time? So yeah, we just compared, they're just like swimming culture, we just like take them out and spit them down, and the adherent culture, we wash a bunch of times and then we like scrape it off into the lysis buffer. But yeah, we haven't looked at population. I mean, we actually have thought maybe even if it's an interesting way to synchronize cells, because if you have like a lawn of adherent and you wash them really well, and then they start producing swimmers and you take them off and analyze them, but we haven't done this as well. Too many things to do.
So I was thinking about your first movie, how even in the Rosette, some movie said swimming off, right? So there's some kind of stochastic events that change the program. So when you add MVP, they don't stick as well. But once they stick, the transition is kind of a big deal, right?
Yeah.
So if you add that after it stick, does it change the newly divided cells program somewhat, or do they have to really shorten, or how does the gel work?
Yeah, we're still kind of analyzing that quantitatively. What I can tell you so far is that when we do the experiments where we adhere them and then add MBD1, those rosettes can make swimmers, but those swimmers can't stick because the drug is still around. So that direction for the attached to swimmers also seems fine in the presence of the compound. It's just it's going from swimming to attached seems to be compromised.
But one that's stuck, and then the newly divided cells from that point, it's still sticking.
Yes, they can still stick. Yeah, they still remain. And we don't understand, if it's stochastic. I mean, what we can see is at least from the movies you've analyzed, it always seems symmetrical. Like if a cell divides, those two cells would either both be attached or they both swim away. We don't see a lot of asymmetrical division events, which is interesting. So it seems like the fate is kind of decided during cell division. They're either both going to be attached or they're both going to swim.
I don't really know how relevant like co-infection model for insect could be with like parasite species. Do you think there's any kind of like competitive advantage, I guess, of the appearance relative to
Right, yeah, it's interesting, yeah, if there's kind of other things, yeah, if they're taking up space in the gut, that then wouldn't be available to other things. Yeah, I mean, we haven't done any kind of co-infection of fasciculata and say Plasmodium, for example. I mean, Plasmodium is usually invading the O kinase going to the midgut, not the hindgut, so maybe it wouldn't matter. But if there are some things that the Confidia are like triggering the immune system a bit, and that decreases the success of the Plasmodium, that could happen. Or you know, viruses, you know, things. I mean, it could be that there are even subtle changes in the immune state of the mosquito might affect other things. We don't really know.
so we're all, a lot of us are stuck on the sticking, so I'm gonna ask another sticking question.
Okay.
And that is, it seems like you only see monolayers, and whether or not that's true, why don't they stick to one another?
That is a really good question. Why? Because I actually really expected them to stick to one another. Yeah, I don't have a really good answer for that. The only thing I can say is like sometimes in vivo, what people have observed is you will have things that look like not monolayers, but when they do the EM sectioning through, some of them actually have longer flagella that still snake down and reach down to that surface. So there's, even though there's multiple layers of them, it's like the ones down here have shorter flagella and the one up here have longer flagella. So there's something about that, yeah, that flagella to surface interaction that's important. And it seems like it needs to be a non-living surface, because even in the hindgut, it's lined with chitin, so they're not really interfacing with an epithelial cell per se. It's got a layer of chitin, and then there's a waxy like lipid layer even above that it seems to be there adhering to. I was going to say something else about that. Oh, there was I probably can't go back now because it's too far, but there are structures that hold the flagellum to the flagellar pocket, which are a kind of more cell-cell interaction, and people have postulated that they might be made of the same thing. So you have these little stapley guys here. So this is the flagellum, and then this is like the flagellar pocket. So this is like the cell body, you know, kind of around the flagellum. And this, you know, people have found proteins that are here. Whether those proteins are the same as these proteins, we don't really know. But this is more of a, so you can see these proteins are on both sides for here, whereas here we really only see the proteins on one side. It's only on the flagellar side. We don't really see any modification of the layer on the other side.
Wait, another round of applause for amazing class. Thank you, Sarah. Thank you everyone for coming in person and virtually, and hope to see you at the June meeting. Good night.
Thank you so much. That's absolutely incredible. Thank you.
Have the students and Megan take a picture. We need a newer picture.